Hello dear student, in last class I have already discussed about different receptors and what are the definition of receptor, what are the terms that is required you have to know about receptor, how they are working in our body, what is their mechanism of action, and different term that is connected with different receptor. And also we studied about classification of receptor, different criteria based on which we are classified or we are differentiated different receptor. Like that, today we are going to discuss about classification of receptor based on their mechanism, that is transduction mechanism. According to the transduction mechanism of receptor, that receptor are classified into 5 types. They are G protein coupled receptor, enzyme linked receptor, receptor that helps in expression of different gene, ion gated or ligand gated channel receptors, and Jack state receptor. In that, in our body, what are the receptors are present? They are most of the G protein coupled receptor, and other receptor also there, and receptor which are using or helps in expressing gene, they are also very important. So today we are going to discuss one by one about the classifications. Today the first topic is GPCR, that is called as G protein coupled receptor. So now we have to study that thing. So in GPCR, what is about G? Here G is guanosine. We have already studied guanosine is a protein or nitrogen base that is present in our DNA also. Here also guanosine is connected with that receptor, that's why they are called as guanosine protein coupled receptor or GPCRs. And most of the receptor present in our body comes under the GPCR classification. So what is GPCR? How it looks like and how it acted to be like. So we have to remember in GPCR they are structured into different membrane. In that they are classified into 7 alpha membrane. So that is also called as 7 alpha or 7 helical structure or 7 alpha helical member or helical membrane plus a receptor. So that receptor has 7 or 7 alpha membrane, and they have 2 parts. One is extracellular part and one is intercellular part. Extracellular domain also have 3 loops, that is number 1 loop, number 2 loop, and number 3 loop we can classify it as, that are external or extracellular domain. And all that, like that in cell also, which is called as cytosol. In cytosol also intercellular domains is there. In that also 3 loops are there, number 1 intercellular domain, number 2 intercellular domain, and number 3 intercellular domain. And here 7 different alpha helical members are present into that membrane, and with that 3 subtype of trimers are present with that is alpha subtype, beta subtype, and gamma subtype, and that we will discuss one by one when we are going to discuss about the mechanism of action of GPCR or G protein coupled receptor. So the first thing is GPCR. In GPCR, that is the binding protein, and here agonist or drug, hormones, neurotransmitter, whatever are present or whatever work they have to show, they have to add or they have to adding with GPCR in that binding site. Here we are discussing about how drug is acting by help of receptor. So here we will talk about agonists. Agonists, what are they? I have already discussed in the previous class in different term is related to receptor. From there you can know about how and what is the agonist. So first thing, that is GPCR, and now drug is not present or drug is not taken. So they are present into the body. Then how they are going to work without drug? When the drug is not adding, they are present into our body in their normal state or steady state or inactivated state. In inactivated state, what happens? In activated state, that binding site remain unoccupied, so it remains empty or remains free. So that time what happens? That time In seventh number membrane, alpha, beta and gamma subtypes are added, or gamma subtypes are present as trimer with each other, and they are generally binded with GDP, or that we are called as guanosine diphosphate, and that we are talking about the steady state when they are not activated. When the GPCR is not activated, that alpha, beta and gamma timers are also not activated, they are inactive, and when they are inactive, during inactive situation, that alpha, beta, gamma subunits, they are generally connected with GDP, and when they are getting excited or drug is binding with that, then what happens? When drug or agonist is binding with binding site, that time binding sites are coming occupied, so that time binding site is not free. When binding site is not free, what will happen? That drug will binding with binding site in GPCR, and this will send a electrical current, and by that 7 membrane, extracellular loop, intercellular loop, like that loop wise, that whole signal will pass through, and that will pass through at last 7th number membrane, and it will connect with alpha, beta, gamma. When drug is connecting and that electrical transaction or electrical signal is reaching to the alpha, beta and gamma membrane or subunit, what happens? It will activate it, and when it is activated, that GDP will go, that GDP will not be there. That time what will come? That time GTP or guanosine triphosphate will come and replace GDP. So what we have come to know here, when the GPCR or G protein coupled receptor is inactive, that means drug is not added, that time alpha, beta, gamma subunits are not activated, that time they are connected to GDP, means guanosine diphosphate. But when the agonist or drug is added with the binding site, it will send a transaction or electrical current. That electrical current will activate that membrane, that will leads to the activation of alpha, beta and gamma trimers. That time alpha, beta, gamma trimers will not bind with GDP, that binded GDP will go, and in place of that GTP will come, and GTP will occupy the place of GDP when the GPCR are activated. So by that I just explain you how the GPCR or receptor transduction or signaling comes under, and in that also different pathways are there, that is called as G protein coupled receptor pathway. That G protein coupled receptor pathway also is differentiated into two parts, that I am going to explain now one by one. So you have to remember that thing, that is very important to remember, GPCR pathway. Now I will discuss about GPCR pathway. So I have already discussed how that GPCR is going to act, how it is getting activated, how alpha, beta and gamma subtype are going to activated or going to be reacted with GTP or GDP. Now we have to remember about what is the GPCR pathway, how that GPCR pathway worked as. That GPCR pathway can be differentiated into two parts, that is adenyl cyclase pathway, adenyl cyclase pathway, and second one is phospholipase C pathway. So that two pathway you have to remember in GPCR pathway. So GPCR pathway can be classified or can be distributed into two parts. One is adenyl cyclase pathway, second one is phospholipase C protein pathway. So before going to the pathway, you have to remember different protein that is going to help in that both pathway. They are GI, GS, GQ and G0. What is the means of I? That I means inhibition, S is synthesis, that is going to activate, that I am going to discuss when I will discuss about that pathway. So you have to just remember what are the receptors or what are the proteins helping that both pathway, that is GI, GS, GO and GQ, and what are the pathway, that is GPCR pathway, and GPCR pathway is classified into adenyl cyclase pathway and phospholipase C pathway. So first we are going to discuss about adenyl cyclase pathway. So now we are going to discuss about adenyl cyclase pathway. What is adenyl cyclase pathway? You have to remember adenyl cyclase pathway is the activation of acetyl or acyl cyclase enzyme. So how it is going to act? That is generally synthesis or generally activated by GS protein and generally inhibited by GI protein. So what it will happen when GS going to activate, that will activate adenyl cyclase protein. When that adenyl cyclase protein is activated, what it will do? It will help in transferring ATP to convert into cyclic adenosine monophosphate. So when the GS is activated, that will activate adenyl cyclase enzyme. That adenyl cyclase enzyme will convert ATP to cyclic adenosine monophosphate, and it is going leads to the activation of CAMP or adenyl monophosphate cyclase dependent kinase activation, and that CAMP kinase activation will leads to phosphorylation of certain enzymes. So what is that adenyl cyclase pathway? In adenyl cyclase pathway, it will be activated by GS protein and it will be inhibited by GI protein. When the GS protein is activated, that ATP will converted into cyclic adenosine monophosphate, and cyclic adenosine monophosphate, it leads to the activation of cyclic adenosine monophosphate dependent protein kinase, and that protein kinase is going to help into the phosphorylation of certain enzymes present into the cytosol. So by that I have discussed about adenyl cyclase pathway. The next pathway is phospholipase C pathway. What that phospholipase C pathway will do? In that phospholipase pathway, that pathway will be activated by GQ protein. What it will happen? When GQ protein is activated, that will lead to the activation of phospholipase C enzymes activation. The phospholipase C activation will lead to the activation of phosphatidyl inositol phosphate enzyme activity or activation. that activation of phospholipase C will lead to the activation of phosphatidyl inositol phosphate activation, and that will lead to the cleavage of phosphatidyl inositol phosphate into two parts, that is diacyl glycerol, that is also called as DAG, and another one part will be inositol triphosphate, that is also known as IP3. What IP3 will do? IP3 will activate the release of calcium into the cytosol, that will lead to the activation of bone and lead to the activation of enzyme. And that DAG, what it will happen? It will increase the activation of PKC, and that PKC, what it will do? That PKC will help into the phosphorylation of certain enzyme. So what is the PLC or phospholipase C pathway? In that phospholipase C pathway, the GQ protein will activate phospholipase C protein activation, that leads to the cleavage of phosphatidyl inositol phosphate into two parts, that is diacyl glycerol or DAG, and IPC, that is inositol triphosphate. That inositol triphosphate helps in the release of calcium ion into the cytosol, that is going to help in activation and strengthening of bone and enzymes, and DAG will lead to the activation of PKC enzyme. That PKC, what it will help? It will help in the activation or phosphorylation of certain enzymes present in cytosol. So by that I have already discussed about the GPCR receptor and what are their mechanism of action, what are GPCR pathway. In GPCR pathway, what are the two pathways, that is phospholipase C pathway and adenyl cyclase pathway, and then we will discuss about what is PLC pathway and what is adenyl cyclase pathway, and then adenyl cyclase pathway we studied how it is going to work, and PLC we studied how that is going to cleavage and going to form IP3 and DAG, and that leads to the activation of calcium ion and phosphorylation of different enzymes. So by that I have easily and very shortly explained about the GPCR and GPCR pathway. In the next video I am going to discuss about ion channel or ligand gated receptor. For today, thank you.